Hello, Alexia. Thanks for you accepting this invite for participate this space for discuss science here in the Brazil. It's my pleasure today present the Dr. Alexia Dubuc. She's in this moment working in the Leibniz Institute in Germany. And today she's explaining more about the mangroves. And I want to thanks, Alexia, for your presentation. And well, the room is yours. Thanks.
Thank you, Juan Pablo. And thank you, everyone, for joining today. So my name is Alexia. I recently completed a postdoctoral fellowship at the Center for Tropical Marine Science in Germany, and where I worked on special variations in fish assemblages across the tropical Eastern Pacific coast, together with Juan Pablo. So that's how I got to meet Juan Pablo. And I really appreciate the opportunity to talk today, especially that this is a special month for breast cancer awareness. And I really appreciate the gesture from Juan Pablo to organize these talks, especially for women this month, in this special occasion. So today I'm going to talk about some studies that I did during my PhD that I completed last year at James Cook University in Australia under the supervision of Marcus Sheaves, Nathan Waltham, Cyril Marchand, and Ronald Baker. And during my PhD, I mostly looked at the utilization of mangrove habitats by fish, and I especially focused on the impact of hypoxia. So first, I'm going to start with a general introduction on mangroves and fish and hypoxia. So mangroves are recognized to be important fish habitats, and this is mainly for three reasons. The first one is that they act as feeding grounds because of their high primary and secondary productivity. The second reason is because they serve as refuges thanks to their prop roots that provide structure for many species, preventing them from being attacked by predators, but also because of the high turbidity of the water, decreasing the visual acuity of these predators. And the second reason is because they act as nurseries for many species that use them during their early life stages. However, the way mangroves are used actually vary incredibly across locations, and as more and more studies are studying how fish use mangroves, they really show different degree of utilization. And this is due to the fact that mangroves have a very large distribution, and they occur across a very wide range of factors, such as, for example, the setting. So they can be estuarine mangroves or coastal or island or bay embayment, but also to their connectivity to other key habitats such as seagrasses or coral reefs, the climatic zones, the tidal regime, or again, the species of mangrove trees that is dominant. The combination of all these different factors provide mangrove with specific attributes that would determine the values and services that can provide. So, for example, here are two pictures of very distinct habitats. The first one is on the left is tidally driven areas in the Indo-West Pacific, and on the right is a mangrove system in the Caribbean. So, as you can see, these two systems, even though they are both mangroves, can potentially provide very different services. Contrasting results in the literature about how fish use mangrove mostly come from comparing results from two different regions where mangroves occur. The first one is the Atlantic Eastern Pacific, and the other one is the Indo-West Pacific. Most of the knowledge initially comes from the Atlantic Eastern Pacific, mostly in the Caribbean, and has been transferred to the Indo-West Pacific. However, the hypotheses I previously mentioned have been applied to this other region in the Indo-West Pacific, but usually it lacks of evidence, and this contributes to providing contrasting results. The main difference between these two regions is the tidal regime. So in the Atlantic Eastern Pacific, the tidal regime is globally microtidal, while in the West Pacific, it varies a lot. So ranging from micro to macrotidal regime. And indeed, a study by Igulu et al. In 2014 identified tidal regime as one of the most important factors driving how fish use mangrove. For example, in the Caribbean, the tidal regime is small, so mangroves are always inundated, providing access to fish constantly. While in tidally driven areas that you can find in the Indo-West Pacific, mangroves are only accessible at high tide, while at low tide, they have to move out. On top of giving like a very important physical constraint, tide also is responsible for many changes in water quality, such as pH, salinity, temperature, turbidity, and dissolved oxygen. Among them, dissolved oxygen might well be one of the most important. As a general rule, dissolved oxygen is scarcer in water than in the atmosphere, making it a perpetual challenge for fish to acquire enough oxygen. And for that, oxygen is considered as one of the most important limiting factors for aquatic life. Additionally, mangroves are one of these ecosystems that naturally experience hypoxia. And this is due to the fact that they have a high production of organic matter by their lithophile. They occur in tropical and subtropical areas where temperatures are very high, and the lack of aeration processes because of their sheltered nature. Moreover, in tidally driven areas, dissolved oxygen decreases during decreasing tide. And this is due to a process called tidal pumping. When the tide decreases, it creates a pressure gradient, and the water that has been infiltrated, the sediment, and that is completely depleted in oxygen because of bacterial activities, is drained back to the water column, creating a sharp decline in dissolved oxygen, and that can cause hypoxia. Hypoxia is a severe issue for fish as it leads to physiological and behavioral changes, but also to long-term effects such as growth, reproduction, and activity reduction. Can also lead to unsuccessful recruitments, perturbations in community dynamics, and ultimately can lead to death. Several studies have looked at dissolved oxygen levels in mangrove habitats, and the studies have recorded dissolved oxygen below 5% saturation, and these low values were recorded almost daily. These low values were associated with low tide, confirming the hypothesis that in tidally driven environments, hypoxia can be a very limiting factor for fish. To avoid mass mortalities that are usually associated with the development of hypoxia, fish have different strategies to deal with these stressful moments. So the first most common adaptation is to move away from these harmful dissolved oxygen levels and to adjacent habitats that might be available. However, this is not always the case, and some species also decide to remain at these oxygen levels. And in these specific occasions, they can use adaptations such as surface respiration, air gulping, or physiological adaptations. In mangroves, we have very limited knowledge on how fish deal with oxygen levels, and we don't know how hypoxia prevents fish from using these habitats or drive tidal migrations. So this is what I looked at during my PhD. I investigated the relationship between changes in dissolved oxygen and fish utilization of mangrove forest. This is a study that was published in 2019 in Biologia. This study was conducted in New Caledonia, which is a small island in the Pacific. It's a French island where I'm actually from. So here you have a picture of the study site. It's a beautiful site that is a mangrove/coral system. So as you can see on the picture, there are some huge coral formations. They are growing right next to this mangrove shore. And I extensively sampled this relatively small scale study site by deploying dissolved oxygen and depth loggers on the edge of the mangrove forest together with underwater cameras to be able to follow variations in fish assemblages as well as dissolved oxygen levels. I sampled the area over several tidal cycles, days, at different time of day, different so at spring tide, neap tide, so pretty extensively. And what I observed is that indeed there was a high correlation between depth and dissolved oxygen, showing that dissolved oxygen values reached very low levels, here a minimum of 13% saturation when the tide was low. The red boxes on the figure show when I actually deployed underwater cameras to monitor fish assemblages at the same time. And I recorded a minimum of 35% saturation in oxygen. So that represents still very harmful levels for a fish. So I wanted to know if there was a variation in fish assemblages across this dissolved oxygen cycle. So firstly, I looked at differences in fish abundance. So I showed that indeed there was a significant differences in fish abundance across the different levels of oxygen, and that the maximum level of fish abundance was not reached until dissolved oxygen levels were above 70–80% saturation. Next, I looked at species-specific responses to these dissolved oxygen levels. And out of the 72 species identified in this study site, I noticed that there were three main patterns that species followed based on the oxygen levels. The first one were species that didn't apparently avoid any dissolved oxygen levels, so their abundance was relatively constant across the dissolved oxygen cycle. And that represented about 53% of the taxa identified. The second group were species that were constantly absent once dissolved oxygen levels were below 50–60% saturation. And this represented about 19% of the taxa. And finally, there was another group of species that had a seemingly high dissolved oxygen avoidance because they were consistently missing once dissolved oxygen levels were below 70–80% saturation. And that represented about 28% of the taxa. Secondly, I looked at differences in relative abundance of individuals within each of these three groups. And I found that the group of species that showed no dissolved oxygen avoidance had also the highest number of individuals, highlighting that perhaps being able to access mangrove habitats, even at low dissolved oxygen, provided some kind of favoring this kind of species. So the take-home messages from these results is that dissolved oxygen does vary with tide and also reaches harmful levels in the specific mangrove system that is tidally driven. In response to these dissolved oxygen fluctuations, fish undergo species-specific migrations. The taxa that were able to access mangroves, even at low dissolved oxygen, were more abundant, had more individuals. And the suggestion from this result is that this tidal migration that I observed could be explained by differential tolerances to hypoxia. And that's precisely the next step I took during my PhD. I determined if there was a link between tolerance to hypoxia and the way the species were using mangroves. This is a paper that is currently in resubmission process, and so the results are not published yet. So what I did is that I used the previous results from the field showing those 3 different patterns of utilization. And I hypothesized that the species following the first pattern had a high tolerance to hypoxia, that the second group could have a slightly lower tolerance to hypoxia, and that the third group could have a substantially lower hypoxia tolerance. And to test this hypothesis, I selected species in each of these groups, and I caught some individuals from the wild, and I brought them back into the lab. So I had 4 study species. The first one was a rabbit fish. I had a bream, a bannerfish, and a butterfly fish. I used a laboratory experiment to determine their hypoxia tolerance. And I used intermittent flow respirometry to do that. So basically what this is a respirometry chamber that you see here on this diagram, in which the individual fish is placed, and the chamber is connected by 2 different pumps. The flush pump is intermittently on and off. So when it's on, the water from the surrounding tank replenishes the water inside the chamber. And when the flush pump is off, the chamber is sealed, so the water doesn't go anywhere. And there is a recirculation loop on which a dissolved oxygen probe is attached that allows to measure at any time the dissolved oxygen inside the chamber. And what we observe is a decline in the oxygen in the chamber that is due to the respiration of the fish. And this is also directly linked to its metabolism. So the experiments were run for about 3 days. And during the last 6 hours of the experiment, some nitrogen gas was bubbled in the surrounding tank to artificially decrease the dissolved oxygen levels, but in a way that it accurately represented what I observed in the field. So in that way, I could be ecologically relevant. So here is a little graph to explain to you what kind of results we can get from these experiments. So on the y-axis is the dissolved oxygen levels in the surrounding tank. And on the y-axis is the rate of oxygen consumption that is linked to the metabolism of the individual fish. So the first step is to determine the standard metabolic rate, which is the minimum rate of oxygen consumption that is needed for the fish to sustain itself, meaning that there is no further activities such as swimming or digestion or stress or all these kind of things. And that value is determined during the first 2 days of the experiment, where the fish is left undisturbed at fully oxygenated water. So in the last 6 hours of the experiment, the dissolved oxygen was artificially decreased in the surrounding tank. And what we observe is something like this. So in the first phase, the rate of oxygen consumption from the fish is maintained relatively constant, despite the fact that the oxygen available in the surrounding is decreasing. After a while, the rate of oxygen consumption will start to decline until a point where the rate of oxygen consumption of the fish is equal to its standard metabolic rate. At that point, we reach a threshold that is called the critical oxygen level, that is usually used as an acute hypoxia threshold. That means if dissolved oxygen levels continue to decline under this threshold, then the survival of the fish is compromised. So for the experiment, I kept going a little further because fish in the wild can be exposed to oxygen levels below this threshold. Maybe they can get stuck, for example, in this environment, or they have nowhere to go. So the dissolved oxygen levels were declined further until we reached the incipient lethal oxygen level. which is a proxy for their resistance to hypoxia, so how long and at what kind of levels they can remain until basically they die, because this is a near-death threshold. So at this point, a fish will flip upside down, so loses its equilibrium, and at that point, the experiment was stopped, and the fish was placed in a fully aerated, oxygenated tank to recover. And for your information, all individuals recovered after the experiment. So thanks to these two measurements, I tried to get a global picture of their hypoxia response. And what I found is that the rabbitfish was the most hypoxia tolerant compared to the other 3 species. So it had a significantly lower critical oxygen level, which was also the case for the incipient lethal oxygen level. So the second measurement, when the fish loses its equilibrium, the rabbitfish had a significantly lower one than the bream, and the bream had a significantly lower value than the other 2 reef fish species that did not differ from each other. So to summarize those results, I found that the rabbitfish came first place in terms of responses to hypoxia. So it was the most tolerant to hypoxia and also the most resistant. And interestingly, the rabbitfish was part of this first group of fishes that showed no apparent disoxygen avoidance. Second came the bream that had a slightly lower hypoxia tolerance and resistance. And again, interestingly, it was following the second pattern. So when fish were avoiding mid-dissolved oxygen levels. And finally, the 2 reef fish species that didn't differ from each other, but had slightly lower resistance to hypoxia compared to the bream, were both following the same pattern, which was the last one, that species that showed a high dissolved oxygen avoidance in situ. So these are very interesting results because it seems like there is a link between hypoxia tolerance and the way the fish use mangrove systems. And it could also suggest that being hypoxia tolerant, like the Siganis, could be a prerequisite to extensively use mangrove habitats, because the species was, for example, very abundant in the forest and all the time there. So I just wanted to give a bit more insight into the ecological implications of those results. So here I made a little conceptual animation to summarize what kind of new knowledge this study can provide. So in the way fish use mangroves. So for example, here, let's pretend we're at high tide during the day and the oxygen levels are above 80% saturation. Then the fish assemblages could be quite diverse and species and quite a lot of individuals on the mangrove edge. As tide starts to decrease, also is dissolved oxygen that can reach maybe levels about 70% saturation, at which point we could see the most sensitive species starting to move out from these habitats. As the tide continues to decline and maybe is associated with nighttime, where dissolved oxygen levels are even lower, then it could reach levels of about 50% saturation, at which moderately sensitive species could also start to move out from these systems. As tide continues to decline and dissolved oxygen reaches levels maybe even below this 30% saturation, as that's been observed, unfortunately not within my study, so I don't have actual fish data to back up this hypothesis, but it is highly probable that even the most tolerant species would have to move out from these systems, or if they remain, would have very specific adaptations, physiological adaptations, or behavioral adaptations, such as the gobies, for example, that are able to do air breathing. So this study shows that there is also a high temporal variability in fish assemblages that are using mangroves. So this provides new insight into explaining the contrasting results that can be observed between those two regions, for example. So not only mangrove utilization is spatially variable because of environmental variability, it has also a high temporal variability. And this is probably even more true for systems that are tidally driven. Sorry. This is something that really needs to be taken into consideration, because depending on when and where you sample an area, even maybe the same site, the fish assemblages could be completely different. So this aspect of very high temporal variability, it's really important to take into consideration when assessing fish assemblages in these highly fluctuating environments. So these studies have shown that fish using mangrove habitats can deal with pretty extreme dissolved oxygen levels. However, they are still intensively used by some species that are very adapted to be there. But there is now an issue, and this is called ocean deoxygenation. And a recent study by Bridberg et al. In 2018 has estimated that the open ocean has lost about 2% of its total oxygen in the past 50 years. And this is mostly due to human activities leading to nutrient enrichment and also climate change. And mangroves are very sensitive to anthropogenic impacts because they are located on the coast where all the runoffs are dumped. So, sorry. We really need to enhance our knowledge on how dissolved oxygen fluctuates in these environments. And this will provide new knowledge and important knowledge for environmental managers and policymakers that will be able to maybe take actions and ensure to the sustainability of these very important systems. So again, I wanted to thank you all for attending today, and again, give an applaud for all these women around the world, and also women fighting breast cancer. Yeah. And thank you, Juan Pablo, for having me.
Thanks, Alexia, for your presentation. It's very interesting, your different approach here for mangroves. And well, now I will speaking in Portuguese for the people who stay here in this moment. Pessoal, desculpa, minha conexão está um pouco ruim, eu não estou conseguindo ativar minha câmera de volta, mas agora está aberto o espaço para as perguntas. Já tem algumas perguntas aqui que eu já vou ler para Alexia. Alexia, this moment is open the space for the questions, and this moment you have 2 questions in the chat. I will read this question, and maybe you can answer these questions. Thanks. Well, João Pedro Coelho question: many mangroves areas here in Brazil are affecting for seed pollution, domestic and industrial. Answer these questions. Thanks. Well, question: how is contamination affecting for seed pollution, domestic? Sorry. How is contamination affecting for seed pollution, domestic?
There's an echo.
Yeah, I know. Maybe it's my computer.
Yeah. Here. Sorry, can you repeat the question again? Because
yeah, of course. Many mangroves areas here in Brazil are affecting for seed pollution, domestic and industrial types. How this contamination can affect the results of the oxygen and different pollution of the fish?
Yeah, so that's a very important point indeed. So in my case, for example, I only sampled a natural system, but I imagine that in impacted systems, the dissolved oxygen cycle will be even more exacerbated. So you could have very, very high values, maybe above 100%, like 140%, 150%. That would not be surprising. But at night, when this photosynthesis stops, then it could have very extremely low dissolved oxygen values, maybe even reaching below 5% saturation. And at that point, there are very, very few species that will be able to tolerate these oxygen levels. So I would say that either on the long term, species could maybe stop using these habitats if they're not well adapted. Or you could see some dominance from very specific species, such as the gobies, the blennies.
Okay, thanks, Alexia. You have another question here for the Cristiano. What kind of the physiology adaptation route along these 5 fishes in the mangrove system?
So there are multiple physiological adaptations that can occur. So it's mostly, so there will be, I guess, 2 pathways in which fish can physiologically adapt. So the first one will be to extract more oxygen from the inside to the outside, from the outside to the inside, sorry. And the other physiological adaptations will be to facilitate oxygen carrying capacities. So, for example, you could have an increase in affinity in the blood of the fish with oxygen molecules that will kind of allow to transport better the oxygen from one organ to another. You could have an increase in some specific cells in the gills that could allow to extract more oxygen. So yeah, there are many of them. That's really something I would like to explore further because yeah, there is a lot of potential and very little knowledge on how these physiological adaptations develop and how short in time they can develop.
Perfect. Alexia, I have one question with you. And you now exploring, for example, the change for the body size in different species between different levels the oxygen.
No, that's not something I have done, but yeah, that's definitely something that would also be very interesting. I don't know if there is really a trend on that, but yeah, that's definitely something that could be explored.
Yeah, well, pessoal, mais perguntas aqui para Alexia. Alexia está aqui, ainda temos bastante tempo para mais perguntas. Well, I have another question. In your experience, you weigh change in the functional diversity or another functional index between different levels or not? What is your opinion about this point?
Yeah, that's now I'm working about that. That's really something I honestly didn't really think about when I was doing my PhD, but now that I have started to get into it a bit more and get a bit more knowledge about this differential functional diversity, that's really something that could be interesting. I think there is one paper that was published not so long ago looking at functional diversity. Differences in functional diversity in mangrove assemblages, but I don't think anything has been done about temporal variability in functional diversity. And that's really something I'm discussing intensively, I guess, in the paper that is in the resubmission process, because that's really a key aspect to ecosystem functioning. So if you have this fish moving in and out constantly from this system, you can imagine that the functional diversity will also drastically change between tides, or I mean across the tide at that point. So in terms of ecosystem functioning, this has very high relevance.
Sorry. People, another question for Alexia today? Well, Alexia, thanks again for your presentation. It's interesting your point, and you understand more about the mangroves. It's very important ecosystem in the world. And thanks for accepting my invitation.
Of course, yeah, it was a pleasure. And yeah, thanks for organizing that. And I was happy to participate.
Ok. Pessoal, o vídeo, para quem não conseguiu assistir ou vocês queiram mandar para outras pessoas conhecidas, vou ficar no canal do CEBIMar. E eu quero já convidar vocês para o nosso próximo CEBIMário, que vai acontecer a próxima quinta-feira, onde teremos a presença da doutora Letícia Lotufa, aqui da Universidade de São Paulo, falando sobre substâncias anti-câncer obtidas a partir da diversidade marinha, desafios e perspectivas. E com ela fecharíamos este mês de outubro contra o câncer de mama. Agradeço a todos a presença. Sorry, desculpa, tem outra pergunta. Sorry, Alexia, is the confusion in the idioms here, the language. You have another question here for the João Pedro Coelho. This is the northern of Brazil, has large mangroves areas and exceeding high tide amplitude. Do you think these specific areas have similar oxygen and phase variation?
So, yeah, I know Brazil is kind of outside the map when I was showing the Atlantic Eastern Pacific region, because indeed it has quite high tidal range. And I know some studies have been looking at tidal migrations, I think in Brazil. I'm not aware of any study that has looked at diesel oxygen levels, at least on the short temporal variation scale. But I would imagine that the results will be quite similar to what I presented. And I would really not be surprised if also diesel oxygen will be quite limiting in these environments. So that's really something that is worth studying, I think. And that would be really cool to kind of bridge the gap between how different tidal ranges can lead to different diesel oxygen levels and influence fish assemblages.
Okay. Sorry, Alexa, you have more questions here. My internet is not is very low today. You have one question here for the Hobson Pinheiro. How the different diversity levels, Pacific versus Atlantic, can affect this fish variation?
How diversity? Like fish diversity?
Yes.
Yeah. I'm not sure I would answer correctly this question. But I know that some sites, some regions in the world have a very high diversity in fish assemblages. I guess that would maybe provide more opportunities for fish, for species of fish to have these high adaptations to oxygen levels, and maybe the resulting diversity in if there is low use of oxygen events could be higher. I'm not sure.
Okay. Maybe Huxon writing more about your question because he's very open.
Yeah,
help
sure.
Yeah.
That's a brand new idea for future studies.
Yeah. You have another question here for the Carlos Eduardo Ferreira. And comparison between Pacific and Atlantic is very interesting approach concerning resistance to hypoxia in fish. Anyways, about the what families is the Caribbean would be more resistant?
Would be more what, sorry?
More resistant.
Oh, more resistant in the Caribbean?
Yeah, for hypoxia.
I don't know. So that's something I was looking forward. It's to find studies that look at diesel oxygen levels in the Caribbean. But so far, I haven't read anything like that. I would say my theory is that diesel oxygen levels will be quite constant in the Caribbean because there is a small tidal range, and therefore maybe species are not used to deal with hypoxia. So I would assume that because species are not used to deal with this kind of oxygen levels, they will not necessarily be highly tolerant.
Yeah, Carlos suggests maybe Aemulidos is one family is more resistant for this hypoxia. I don't
The
know.
immunity. Mm.
Yeah.
Yeah, they were very common as well in my study site. I don't remember on top of my head like that which pattern they were following, like if you follow my results, but I think they were medium tolerant. So avoiding levels of oxygen that were below 50, 60%.
Okay. Alexia, I thinking about your presentation here, and I have one curious about your point. For example, you weight more resistant for hypoxia for a species very near or is sister species in phylogenetic correlation or not?
Are you talking about the two reef fish species?
Yes, exactly. You have, for example, sister species, and maybe this species has similar tolerance for the hypoxia or not. What do you opinion about this point?
Yeah, I guess there is definitely a genetic part in being tolerant to hypoxia, but I also imagine that there is a specific setup for the species, because species can become hypoxia tolerant once they are exposed more frequently to these oxygen levels. So, for example, those two reef fish species that I observed in my mangrove site, they're not usually associated with mangroves, which was quite surprising to find them there. But so in the field, they were avoiding these oxygen levels that were below 70, 80% saturation, which is pretty high. While in the lab, when I tested them, their actual hypoxia tolerance was about 20% saturation. So technically, they could be using mangrove systems at even lower levels that they do, but they might have like chronic effects more easily, let's say, than other species. Yeah, that's really so. I think the genetic is something, but also being exposed continuously to these levels can also trigger some adaptations that they can develop through time and over just one generation. But that's something definitely that they can maybe transmit to the next generation, and kind of build some more tolerant individuals through time.
Oh, perfect. Well, Alex, thanks again. Pessoal, mais perguntas para a Alexia que esteja no chat, que eu esteja esquecendo, por favor. Ou alguma pessoa está com alguma outra pergunta aqui. Bom, pessoal, muito obrigado a todos por a presença. Espero vocês na próxima quinta. Again, thanks, Alexia, for the participation in this space.
My pleasure.
It's very important for sharing your results here. Thanks.
Thank you so much, and thank you all for your interest and time. All right.
Bye-bye, people.